Welcome to the LabRoots webinar, where we're presenting Dr. Mathias Uhlén from the KTH Royal Institute of Technology in Stockholm, where we'll be presenting next-generation pan-cancer blood proteome profiling using proximity extension assay. My name's Dale Yuzuki. I'll be your moderator today with a short introduction about Olink. Olink, if you're not familiar with it, is a platform based around technology called proximity extension assay. It uses two antibodies and a covalently linked DNA oligos to increase specificity of protein detection. The beauty of the assay and technology is that we're able to multiplex from the dozens to the thousands of protein analytes from a single sample. In addition, right, you're able to use very small amounts of sample down to 1 microliter, depending on the level of multiplex, and in a variety of formats. As you can see on the right, we have Olink Explore with 3,000 protein targets, Olink Target, which are panels of 96 or 48 protein targets. We have some 15 panels pre-configured in that way. Olink Flex, which is our newest mix and match product, where you're able to choose 21 targets out of a menu, a preset menu of about 200. And Focus, which is a fully customized product, again, with 21 markers. In addition, we have Olink Insight, which is a data platform for biomarker analysis. And we'll have a little bit more about Olink Insight later in the presentation, because some of the data actually from this presentation is on that particular platform. Again, the aspect of the PEA technology is we were able to go from discovery to clinical translation on a single technology, on a single platform. The Explorer is the 3,000 targets, like I mentioned, Target with the 96, and then Flex, which is a mix and match semi-custom panel of 21 markers. And lastly, just to point out the benefits of the particular technology is very, very high sensitivity in terms of a multiplex assay, exceptional specificity in terms of knowing exactly what you're measuring, minimal sample volume, like I mentioned, 1 microliter for Target 96, 6 microliters for Olink Explorer, and a very wide dynamic range in terms of being able to have high sensitivity, high specificity, and being able to measure many logs of dynamic range. Without any further delay, I'd like to go ahead and introduce Professor Mathias Uhlén. Like I mentioned, he's at the Royal Institute of Technology, otherwise known as KTH in Stockholm. He's the founder of the Science for Life Laboratory, which is a Swedish government sponsored effort and one of the largest European laboratories of its kind. And also since 2002 or 2003, it may have been a typo, he founded the Human Protein Atlas Project. Now, this has led to over 500 publications and has spun off several other atlases since. As you can see here, it led to the Cell Atlas in 2017. It led to the Tissue Atlas in 2014, the Pathology Atlas, and a Blood Atlas. This landmark 2015 paper was one of the most cited in Science, where Dr. Uhlén is the first author. And today he'll talk more about the Human Disease Atlas, which was the most recent one launched. Okay, and I think with that introduction, I'll hand over the presentation to Dr. Uhlén.
Thank you very much, Dale. It is a pleasure for me to give this presentation about one of my favorite topics, and that is to talk about next generation blood profiling. So with that, then, I just want to say a little bit about myself. I am actually not I have about 800 publications. I am, as you say, the director of a Human Protein Atlas Consortium. So with that, I just also want to mention the Science for Life Laboratory, which I'm very proud of. It's a national infrastructure here in Sweden, which is big data, technology-driven research, and we are actually now in Stockholm 1,200 researchers. So what I would like to start with is to give you an introduction to the Human Protein Atlas. This is an effort to create an open access knowledge base for all human proteins, basically. And what I would like to do in this presentation then is first to give you an idea, an introduction to the first 20 years of this program, then move into the theme of this presentation, which is the next generation blood profiling, then talk about cancer profiling, and then end by saying a little bit about how you can access the data, which is now an open access resource. So first, a little bit about the Protein Atlas. It is one of the most visited databases in the world, I should say biological databases, and it is selected to be a core resource in Europe for biology. We have a lot of data in the Protein Atlas. We have more than 5 million web pages, which are updated annually. And we also have more than 10 million annotated tissue images. And as I said, we have now published more than 800 publications from the consortium. So the flagship paper from the group or from the program was the Tissue Atlas that was first published in Nature in 2014, followed by the Science paper in 2015. And this has then been followed by some other major launches, as was mentioned by Dale, the Subcell Atlas in 2017, a Cancer Atlas, a Blood Atlas, a Brain, and then in 2021, we moved into the single-cell transcriptomics, and just a few months ago, we then released the Disease Atlas. So so during these 20 years, we have then done a lot of milestones, as is outlined on the right here, but I don't want to go into that in detail, simply to say that we are trying then to answer central questions in human biology. What are the targets for future drugs and diagnostics? How many proteins are in a given cell, and so on? How many proteins are tissue specific, and so on? And the way we started doing that was to build a resource of antibodies to all human proteins. We actually decided then to not go for recombinant antibodies, which are of course excellent for therapy. We are not going mainly for monoclonal antibodies, which is of course excellent for diagnostics and cell sorting, but instead we went for what we called HPA polyclonals, since we are working with tissue profiling, denaturated proteins, and so on. And this is actually what we have used, and we actually we have generated actually close to 60,000 antibodies in this way. So in the version 22 that was launched in December last year, a few months ago, we have a lot of new features. One is that we are actually showing the structure from AlphaFold for all human proteins. We have introduced a new multiplex tissue profiling. We are also showing a lot of more single cell data, and actually included is also data on more than 1,000 human cell lines. The protein structure section shows 3D models for all proteins, both experimental structures from PDB, but also imported predicted models from this amazing project called AlphaFold 2. What is nice about this is that we can also show on the structure the antigen binding site for all our antibodies, so we know exactly where in the structure the different antibodies are binding. So with that short introduction, I want to move into the main theme of this presentation, and that is next generation blood profiling. We are extremely happy about the technology development that has happened in the last few years. A few years ago, we published a paper in Science, 2019, where we had a blood atlas. But what has happened since then is that we now have technologies for actually looking also at the very small abundant proteins in blood. And that has allowed us now to move into precision medicine with a focus on blood profiling, really to start in a completely new way, be able to do early detection of disease, stratification of patients, and monitor of treatments. And I will focus today on the cancer part of that. We are using, as Dale was saying, the Olink Explore. The data that I will present today is on the 1500 version of Olink Explore. And again, this is a fantastic technology where you can do multiplex assays with high precision and very high sensitivity with very low sample volumes. So a few years ago, I guess in 2020, we published a paper where we used this technology to profile healthy individuals that we followed for 2 years. We call it the wellness cohort. And what we showed in this paper that was published in Nature Communication is that each individual that we looked at had a unique but also a stable plasma profile. So everybody that are listening now has his own sort of blood fingerprint, which actually is different from your neighbor. And this is, of course, very exciting. We also moved on and looked at the how many of these protein differences in proteins levels are due to genetics. We call this PQTLs, and published a paper also in 2020 in Genome Medicine, where we actually identified several hundreds of these PQTLs. Then we moved on and we looked in different diseases. And this has been published, so I'm not going to go into it, but we looked at type 2 diabetes patients, published in 21. We're looking at preterm infants with a couple of papers in pediatric research. And we also, of course, looked at COVID-19 patients, which was published in 2021. But what we were very encouraged about the results and all the data you can get out of this amazing technology. So we decided a few years ago to embark on a very bold project to try to create a very detailed and comprehensive map of protein levels in human blood, and try to do that to include most major diseases, create an open access resource, identify biomarkers in this way. What we've done is now, and we are finished, is that we looked at 88 different diseases spanning from cancers, infections, neurobiology, neurodegenerative diseases, autoimmune diseases, and cardiovascular diseases. So really try to cover most of the major diseases in humans, and of course also healthy individuals as a comparison. This just shows you we have analyzed now about 10,000 patients, and we have determined then the blood based on EDTA plasma to try to be able to compare between the different cohorts. And the results for all these diseases will be presented in the Open Access Human Protein Atlas. I come back to this. So the first disease that we looked at is cancer. And the idea then here is to try to see if we can find a blood profile that distinguish cancer patients from one type of cancer from the rest. And also, of course, to distinguish cancer patients from healthy individuals. And this is data that has been done by Maria Bueno Alves and also others in the group. I'm not going to go into the workflow here, but simply to say that this, of course, involves sample collection. We do a lot of differential expression to compare the protein levels between these different cancers. Then we are also doing, trying to create a prediction model using AI-based models. And then also then to find a protein panel, which is ideal, and then classify the cancers, unknown cancers, with this model. The differential expression, we then analyzed all the 1,500 proteins, and I'm not going to go into the details here, but simply to say that there were quite a few of these proteins that went up and down in these different patients. There was overlap in some cases, as you can see here, that especially the lung and colorectal cancers, there was a lot of proteins that simultaneously goes up in both of these cancers or cancer patients. So basically what we are then moved is in a sort of a machine learning mode then to try to then see, can we find an algorithm that can sort through all of this data? It's more than 2 million data points. And the way you do that traditionally is that you have a train set, and then you have a test set, and then you try to find what are the proteins which are most important to predict if a patient has a particular cancer. And this is what we've done. And this is a rather busy slide, but it shows you the results for 12 of the cancers, from leukemia to the left here, all the way to ovarian cancer to the right. And you can then see the population of cancers, the levels when you are predicting with all the proteins that we have analyzed. And in the middle row here, you can see the so-called ROC curve, where you can see how specific and sensitive is the assay. And we are very happy to see that in most cases, at least in the test set, then we are seeing area under the curve of well over 0.95. But you also see in the last row here, this is called a confusion matrix that gives you also the false positives and the false negatives. You can actually see in the red boxes that we are seeing false positive and false negatives. And this is, of course, something that I will come back to in the last part of my presentation. But anyway, since we're using machine learning, we asked for an algorithm to actually, can we scale down the 1,500 proteins into a more convenient panel? And the model then turned out to give us 83 proteins that can then be used to classify patients from all the different 12 different cancers. And I should point out that the big, very frequent cancers are all included here: the lung cancer, the prostate cancer, the colorectal cancer, and breast cancer. So with that, and here you can see, and this you can also see if you go to the website, you can actually click on these and see the results for each one of them. I'll come back to this. Just to give you a little bit of the results, this is now some examples from the protein panel. So you can see on the top left then that the protein CD244 is elevated in acute myeloid leukemia. You have another protein, PAEP, that at least according to us is increased in ovarian cancer. You have a very nice elevation of GFAP, GFAB, in the glioma patients. And on the bottom row, you can see SLAM-F7 in myeloma. You see elevation of the PRDX in colorectal cancer and so on. So, and this you can of course also see in the open access resource. I come back to this. So with this, we are also analyzing the classification using this model and the panels. And here you can see the area under the curve in the rock curves for the 12 different cancers. If you use all the proteins, which is in blue, if you use the protein panel, which is green, and if you only use the top 3, which is yellow, and if you only use the top protein in red. And what is very apparent is that in all cases, you get much better sensitivity and specificity if you're using the panel as compared to use a single biomarker. It's also, of course, interesting to see, can we use this to pick out which of the patients have cancer, and not only then, as I showed before, compare it with other cancer patients, but also to compare it with healthy individuals. And this just shows you 4 examples of the comparison between the healthy individuals, which is called wellness here, and then 4 different cancers. And you can see that there is a pretty good distinguishing between, and the area under the curve is in most cases very, very good. Actually, AUC 1 for the ALM, but or not for the leukemia, but also a little bit less for the breast cancer and the lung cancer. So with that, then I just want to say a little bit about the open access human disease blood atlas. So I hope that you all will go to this. This is then at proteinatlas.org, but we also have the data presented in the Olink Insight that was mentioned by Dave earlier. So in the protein atlas, in the disease atlas, the protein levels across all the cancers are shown. And you can then see, for example, then the FLT3 proteins, which are elevated in acute myeloid leukemia, as you can see here. And this is then, we show the data for all the 1,500 proteins that we have analyzed. There's also the Olink Insight tool, and we're very happy that all the data has also been transferred to the Olink Insight. And there you can actually then go in and look at the in-depth results for a specific cancer type. And I urge you all to explore both of these resources, which are open access. We published this in a preprint, so if you're interested, you can go to that and read about it. It is also in review right now, so we hope to publish it in the future, of course. So with that, I just want to conclude then and sort of summarize and say that we have fantastic new tools for comprehensive and quantitative profiling. There is a possibility, we think, to identify patients with cancer from just a small drop of blood. It says 10 milliliter here, but it should be 10 microliter, of course. And the version 22 of the Human Protein Atlas, which is published, contains then the new human disease blood atlas. So I urge you all to explore the data by yourself. Before I end, I want to just also say a little bit about the biomarker dilemma. This is the fact that false positives are a major issue for population studies. And in this case, we're talking about cancer. They are very heterogeneous, but there's also a sort of a rather noisy biology around these things. So this means that you will then, when you're detecting a true positive, which is symbolized by the orange person here to the right, you will also, especially if you do a screening method, you will actually also identify false positive, which are so symbolized with the blue persons here to the right. So even if you have a very, very high specificity, area under the curve, maybe 0.99, it will still give you thousands of wrongly identified patients in a screening mode, false positives. So this means that if you want to move into the clinical arena, I think it's absolutely essential that you have secondary and independent assays to identify the false positive, then rule them out. So you can then focus on the true positives, but then, of course, identify those and start treatment maybe before you have symptoms, actually. That would be ideal. So with that, I also want to say a little bit about the funding. This is funded by a non-profit organization, foundation here in Sweden called the Wallenberg Foundation. But we also have got funding from other foundations like Novo Nordisk in Denmark, Erling Persson Foundation, the Heart and Lung Foundation here in Sweden, and actually also have funding from the Jean Zuckerberg Foundation. And we are, of course, also very proud that we're part of this EU infrastructure elixir for sharing the data resources in an open access manner. So before I end, I just want to say that this is an effort which has been done by a lot of people during 20 years, although the precision medicine part of this is focused on the last 2 or 3 years when we are getting this fantastic new tools to do it. I want to thank Fredrik Edfors, Linn Fagerberg, Maria Bueno Alves, Fredrik Pontin, especially for all their efforts, but also the other people on this slide. I also want to say that during the 20 years of the HPA, there has been a lot of people involved, and these are just about 700 people that have been formally involved to create this resource. So with that, I want to keep this to about 30 minutes then. I just want to show the site where we are, the SciLife Lab. This is in Stockholm. It's at the Karolinska Institute campus, the medical faculty. And we are at the Science for Life laboratory, which you can see to the far left. So with that, I think I want to thank you for your attention, and I hope that you have questions or comments to my presentation. And then hand back to Dale, I guess.
Thank you, Mathias, for a wonderful talk. Certainly, those of you in the audience, if you have questions, feel free to use the Q&A box to ask your question. Mathias, if you wouldn't mind clicking back to that code for the preprint, I thought that was a really interesting read. If I understand correctly, right, you looked at some 1,500 individuals using Olink Explorer that was able to examine these 1,463 proteins. And by the way, for those listening, if you're interested in that particular human disease human blood disease atlas, go to proteinatlas.org, and then you'll see panels of all the different atlases that they have. And it's just one click away as far as choosing the disease blood atlas. A question that I had for you, as far as cancer, these particular samples were not pre-selected for stage, right? You just grabbed whatever 1,500 samples from a biobank. Can you tell me more about the stage of these cancers and whether or not it would bias sort of the findings?
Okay, no, that's so. So in this case, we're using a biobank from Uppsala. It's called UCAN. And the good thing about this biobank is that it has been collected during 15 years. And we know, of course, we know the stages when they were diagnosed. We also know the follow-up, how these patients have, survived during the years since they got diagnosed.
Right.
And all the blood samples have been taken before they start treatment, which is, of course, very important. We do have stage information, and in the paper that you can see right here, there is also a small part about where we've been looking at stages, because obviously what you would like for cancer diagnostics is to actually be able to identify patients already in stage 1 or even 0, but stage 1. So, and we can see that in at least some of these cases. But of course, when you're doing this pan-cancer, you're including 12 cancers, you can't have too many patients analyzed. So that's why we are now following up this study with another 10,000 patients in order to validate the results and see that what we found. And we will then use the new panel from Olink, which is the 3,000 panel. So we're very excited about this. I should also say that I have focused here on the cancers, but also the other diseases are now being analyzed, and we will release them again in a sort of open access manner when we have them analyzed and checked and validated and so on. So you will then be able to compare the protein profiles in this cancer with the protein profiles of obese people, of people with infectious diseases, or even neurodegenerative diseases.
Sure. And as far as then the findings, right? Naturally, you can't be an expert on all diseases and all the biomarkers of these diseases, but protein biomarkers in cancer were studied extensively in the 90s. I mean, I was one of them, right, working on melanoma in the 90s and just doing, just Western blots on a single protein target. For those who are experts in the cancer biomarker field, were those findings unusual, surprising, or, for example, the colorectal cancer markers? Did it cover sort of well-trodden ground as well as some surprising findings?
They are certainly surprising in some cases, and certainly very expected in some cases. So the main biomarker for glioma, for example, is a very well-known brain protein. There are also for the ovarian cancer, it's a well-known female proteins. Other proteins are expressed or produced in the liver, so this must be secondary effects. And some of the proteins are intracellular, so they are obviously leakage products, probably. So it's a little bit, but it is all. Maybe I should also say at this point that the sensitivity is so amazing compared to what we had 10 years ago. I'm sure that some of you that are listening to this remember the trial, the Theranos trials with Elizabeth Holmes just a few months ago, where she claimed, and the company claimed, that they can do 100 assays from a small drop of blood, and that was, of course, fake. And here we are, and we can do 3,000 assays just 10 years later from a small drop of blood with exquisite sensitivity. And so it is an enormous revolution that is happening right now in this field.
You heard it here first, folks. We're living through a revolution, right? And who less to say it than the person who founded the Protein Atlas way back in the sort of with all the excitement of the Human Genome Project. I mean, in along those lines, right? In terms of such a wider lens that you're looking at, and like you mentioned, these unexpected findings. I really appreciate how you closed in terms of looking at screening applications and false positives and false negatives. And naturally, you have to get disease prevalence in there as well. So it's a complicated set of considerations, right, in terms of positive predictive values and all that. One question, though, came in terms of looking at validating being secondary assays. Would you say in the screening context for cancer, it would be sort of just conventional diagnostic techniques, right, that you would look at someone possibly, right, screened for cancer, and then they look at it via regular diagnostics? Can you comment on that?
Absolutely. So, I mean, fortunately, we do have quite a lot of screening procedures for some of the more common cancers. So, obviously, for if you have breast cancer, you are screened. I mean, at least in many countries, you are screened with mammography. If you have if you look for colorectal cancer, you would have colonoscopy or blood in feces and so on. So the way I see this is that you would probably not screen if you don't have a very sort of good screening methods that can come afterwards. But I think we're quite fortunate that many of these very common cancers, we do have these kind of secondary or screens. So instead of calling in people maybe every 10 years or so, you might then want to do a simple blood test every year to actually select those that should come in for a proper mammography if it's breast cancer, or a lung scan if it's lung cancer, and so on.
Yeah.
So this would then solve this really big dilemma that I talked about, the false positive dilemma.
Well, certainly in the last, I don't know, this is now 2023. So say the last 7 or 8 years with entities like Grail and Guardant and Freenome, looking at ctDNA, other small startups looking at circulating T cells, cancer T cells. CTCs in terms of circulating tumor cells. Dale, get it right. How do you view this in terms of the protein findings versus DNA findings? Do you see room for complementarity there? Or would you say that the predictive power of such multiplex findings, you mentioned 83 proteins, being able to segregate out very cleanly 12 different cancers at once? Can you comment on that?
No, absolutely. That's a very good question. So obviously, if you look at the DNA or RNA-based methods, they are extremely complementary to this. So like GRAID, for example. So I could see that as a parallel assay that you do, but I would still think that if we found a potential breast cancer patients, I would like that patient to go through mammography in order to really establish if you have a cancer or not.
Sure.
But I also want to say that this is just the first study, and we all know about biomarkers and how noisy data is and so on. So even if we have analyzed altogether now 10,000 patients, it is still, when you're actually saying for each disease, it's not that many patients. So we need to continue, and the community needs to do this. And this is one of the reasons why we are putting out this in an open access manner, so people can get inspired and also do their own studies.
What a tremendous resource. I mean, what a remarkable act of generosity as well, right? To make these kind of data sets, some 2 million data points available.
Yes.
And
Yes.
on that note, regarding the HPA project, one question here is absolutely relevant. Do you plan to get into rare diseases?
Yeah, that's a very good question. So the protein atlas has up till basically now been focusing very much on normal, healthy individuals, healthy tissues, blood in normal, healthy. What we are doing now is we are moving into the diseases, but we have focused on the big diseases so
Understood.
far.
Yep.
But it is of course interesting to also go for rare diseases and so on. But the focus right now is to create a resource for the major diseases that can not only be used to get inspiration, what is interesting, but also gives a reference for people that are sort of analyzing a specific type of, for example, rare disease then.
Sure. Now, you mentioned disease heterogeneity, and I know in one of your slides you talked about being able to go into other diseases. And one question came in specifically with regard to neurological disorders, specifically Alzheimer's, and its native heterogeneity, the real importance for blood-based biomarkers. I don't know if you can comment either on the AD example or perhaps give us a hint in terms of what the next sort of disease area would be.
No, but I mean, we have analyzed now AD Alzheimer patients, Parkinson patients, ALS patients, and so on. Also, actually, schizophrenia and bipolar patients. So these will be actually presented. But I fear, just like you're saying, that the heterogeneity will not be much higher than what we are finding for the cancer patients. But actually, we don't know yet. We have the data. We need to analyze it, and we will hopefully then publish the results relatively soon. But then we would like to follow it up with more patients, of course, for these very important diseases.
Sure. One question that is interesting, right, is as far as thoughts on adoption of this clinically, right, in terms of translating it into wider spread testing. Do you foresee that? Are you optimistic or pessimistic? By that, I mean, it becomes a technology, a commercial, regulatory. It's a multifaceted difficulty. And obviously, you're working on the blood atlas, right, for non-invasive testing, right, or minimally invasive testing. And the whole goal is to improve healthcare, right, to improve, human health. What are your thoughts on, its translatability, if I can use that word, of translating into the testing environment?
Well, first of all, maybe I should say that we're collaborating with a company here in Stockholm where you actually take a blood sample at home and you send it by mail in a sort of a dry blood spot sort of format, which allows people to actually then sample themselves at home and send them with mail to the lab and get the results. So this is this, and we have excellent results from this. It seems like it's as good as if you're using the sort of standard EDTA plasma from people. But of course, we would like this to be translated into the clinic. But as you say, there's a long way to go. It's you have to validate. Probably we would like other researchers to join in and actually do different diseases maybe in a more targeted way. What we are doing is this pan disease, which of course is interesting as a sort of basis for this. but it's probably not enough to actually be bold enough to go into the clinic. You have to then validate it in larger cohorts.
Yep.
Also, maybe in longitudinal cohorts, and also then with a lot of healthy individuals.
Yeah.
But saying that, I mean, when I'm looking at the data, and you can all look at the data in this publication, I really think this. I would be very disappointed if you can't translate this or similar into the clinic in the future.
Well,
There's
when you see, yeah, go ahead.
Yeah.
I'm sorry.
There is, especially for cancer, if you find a cancer early on, I can give you an example. Breast cancer, you have 99% chance of surviving if you find it at stage 1. And you have only 20 to 30% chance of survival if you find it at stage 4. And obviously, if we can have this type of screening, find cancers early on,
Absolutely.
I mean, it would save so much cost to society, but also save a lot of lives. So I'm very much hoping that this can translate, but I also know that it's naive to say, is it 3 or 4 years?
Right.
We'll see.
Yep. It becomes a difficult problem, right? When you're looking at, say, pan cancer and looking at so many cancers at once, it becomes a real practical problem. But at the same time, you look at those AUCs that you showed, and you see 1 and 0.99 and 0.98, just say these are remarkably discriminatory, right? Being able to detect cancers, being able to detect, I think the unusual approach in your work, right, was that you were looking for the unique signature of each of the 12 cancers, right? It wasn't just cancer or not, it was each individual cancer. And on the machine learning side, there was a question that came up regarding thresholds, right?
Yeah.
In your machine learning model, it was a data model, right, that combined these 2 million data points and these 1,500 samples and the 1463 markers all into what can discriminate versus not. Can you comment on that in terms of, like, the threshold cutoffs? And I guess it gets back into the fact that you had historical data on patient outcomes with the 15 years of data on these samples. Is that correct?
No, in this case, the algorithm or the machine learning, and this was an algorithm called GlimNet that we use. We also been looking with random forest and others. But what we are basically asking the machine learning is to go to the 70% of the samples and try to find a profile that would identify one cancer from the other 12 cancers as good as possible. And then you're just checking this on the unknown, well, for the algorithm unknown samples, which is 30%. But obviously, it would be now extremely nice to test the algorithm on a very independent cohort. And and it would be interesting to, combine this with the community where people are doing these kind of work, and then we could actually use the algorithms to actually analyze it further and validate the results.
Right. And going back to, for my last question, going back to some of your first slides where you reviewed some of the work from the Scapus longitudinal cohort that you had, what you had PQTLs coming out, you had type 2 diabetes, you had preterm infant birth, and you had COVID. I mean, it seemed like that longitudinal cohort just was a fountain of all kinds of interesting research. Is that work still going on? Do you still have more places to investigate with that longitudinal cohort? And also, what does that imply for other cohorts? By that, I mean the importance of longitudinal wellness testing, so to speak, or wellness sampling and following individuals over time.
Yeah, no, it's very interesting, and as you say, what we are finding in these cases, I'm just going to show you this. No, I didn't show any results from this. It is, of course, in the papers, but the fact is that each one of us seems to have, when we are healthy, a very stable fingerprint of blood. And when we get diseases, and we had, for example, one person that did lose a lot of weight. We had another person that got a serious infection and so on. Then some of these profiles, just goes very different.
Wow.
So I think that what we are seeing is at least signs that it would be very good to maybe do screening, every year or so, and look at the blood profiles and, see, do you have cancer? Do you have other diseases? But again, the false positive dilemma is something that has to be addressed all the time. We are not out to make people, to make people worried. We want to help people, and the false positive dilemma is of course not nice. But the nice thing about longitudinal studies is, of course, that you can compare your profile with your own profile 1 year earlier or so, which of course is also very nice compared to actually comparing your profile with the rest of the population.
Yeah.
So it becomes more individual.
I think what you point out is the real-time nature of proteomic or protein testing in blood, right? Is that it's an accurate real-time indicator of general health. And then if you had a baseline for individuals, right? And like you say, when things start going awry, when signals start to change, how powerful that can be. But
exactly.
Just like you mentioned, so many individuals, so much individual variation, right? That's driving protein determination. How does that all start? But we start somewhere. You're going to say.
But again, I must also stress that sometimes too much information is not good for us.
Yes.
And so one has to. I think one has to be very careful what you're actually analyzing. I'm not a very big believer that in the end you test for everything, because if you test for everything, you will always find something. And if you find something, you have to do something about it. And in most cases, it might not be clinically relevant. So I think it is very important that we know what we are doing in the end and why.
Well, I want to thank you, Mathias, for your generosity in sharing these things today. Certainly, the questions that were not able to be asked, we will address individually via email. And thank you very much for joining us today.
Thank you very much.
Great.